Florida's agriculture industry relies on dependable, high-quality research, and the Florida Crop BMP datasets are a vital resource for researchers. Their value hinges not only on precise measurements, but also on the accuracy and completeness of data entry, such as well-defined variables, detailed management and growth condition, and whether special formats or cell comments were used to convey information that could be lost in further processing. To ensure data integrity throughout development, FDACS and UF-IFAS have developed a quality assurance and quality control, or QA/QC tool that helps researchers validate and refine their datasets. We emphasize the difference between quality assurance, which refers to assessing quality during product development, versus quality control, which deals with evaluating a final product against predetermined specifications. The QA/QC tool contains elements of both QA and QC. In preparing a BMP dataset, researchers are strongly encouraged to add information regularly as experiments progress, and as part of this process, use the QA/QC tool to conduct QA/QC tests on a regular basis. This video describes the use of the QA/QC tool, which is written in R. The script reviews BMP datasets according to 4 criteria: correctness, completeness, coherency, and compatibility, which we call the 4 C's of dataset quality. Correctness refers to whether values are measured accurately and transcribed correctly to the workbook. FDACS strongly urges researchers to review their data carefully prior to entering the data into the BMP template. The QA/QC tool mainly provides simple checks for ranges and means. Completeness refers to whether all types of expected data are included. This includes metadata and information on crop management, soils, and weather, as well as actual research measurements or observations. Coherency concerns whether different worksheets of a BMP dataset can be linked or merged correctly via identifiers. For example, experiment ID and site found under the Treatments tab are linked to experiment ID and site in the Water, Experimental Designs, and Fertilizer tabs. Compatibility refers to whether the dataset is structured in a manner that facilitates processing with different software tools. This includes whether all variables used are defined and have units of measurement assigned. To understand how the 4 C's are interpreted in the QA/QC tool, let's look at an example report drawn from a series of potato experiments conducted between 2011 and 2014. Later, we'll show you how an R script is used to generate this report. Each QA/QC report is designed to be a self-documenting PDF. The introductory section includes an overview of the report and a summary of how it was generated. Section 2 looks at the completeness of the contents and provides summaries of categorical and numerical data for each of the BMP template's 26 data sheets. As an example, let's look at how the report collates the 619 rows of information found in Sheet 02 Yield Summary. First, the categorical data, which here correspond to identifiers experiment ID and site, highlights the values related to each identifier and the frequency with which each value appears. Frequencies can help in checking whether the identifiers have been entered as expected. For numerical values, simple statistics such as the minimum, mean, and maximum are given. Note that the results include identifiers like the study year, plot ID, and treatment number, which should still be checked for correctness. Next, these same variables are represented graphically with box plots, where the red dot indicates the mean and the black bar is the median. If a sheet lacks data, as in Sheet 04 Crop Health, it will be reflected in the report. To help compare the data from different sites during the study years, this graphic was generated to show the timelines for fertilizer applications, plantings, and harvests, or F, P, and H, respectively. The report also shows a map of Florida with any locations described in the dataset. In this case, exact locations of farms are not shown to respect the owner's privacy. We emphasize that these are simple checks for correct values. The main data checking should be undertaken as an essential part of the data preparation. Section 3 examines the coherency of identifiers, comparing identifiers used across sheets to ensure that levels are named in a consistent manner. For example, in comparing the identifier field locations for sheets E2 fields and E3 plots, we see that the levels 18 and 19 are missing in E3 plots. In this case, we know that trials planned for one farm were not executed in 2021 and 2022, so the missing values are expected. The final section addresses the compatibility of the data with other tools and software. A key criterion is whether each variable is fully described. Here the results show that all the variables present in the sheets are in the dictionaries. All the variables are associated with definitions, and all the variables are linked to an ICASA name. Finally, the report checks for formulas, merged cells, or comments attached to cells, any of which could affect the compatibility of the data with other software. Here the report identifies a potential problem on sheet E3 plots, finding merged cells at I1 through P1. A simple inspection shows that the cells between I1 and P1 have been set aside for instructional information, so it can be accepted. Let's run through a demonstration of how a QA/QC script works by examining a dataset of potato experiments conducted in Hastings, Florida. We assume that you understand how Florida crop BMP datasets are organized and are familiar with the basic operation of the R language, use of RStudio, and have some of the packages utilized by RStudio, like R Markdown, OpenXL SX2, Knitter, ggplot2, Maps, MapData, and Reshape2 already installed on your computer. For this demonstration, we've created a working folder named Hastings QA/QC on our PC, into which we've already loaded the dataset we'll be testing, FDAX Hastings Potato 2011 to 2014 demo. Next, in the Florida Crop BMP dataset site on GitHub, in the Tools folder, we'll find and open the subfolder Quality Assurance Control and download the R Markdown script Florida Crop BMP QA/QC single dataset to our working folder. Open the script in RStudio, then specify the name of the dataset to be tested, as prompted here on line 13. We've entered FDAX Hastings Potato 2011 to 2014 demo. This should be the only modification you have to make to the R script. Run the script from RStudio by entering Alt-Control-R or navigating through the menu bar to Code, Run Region, Run All. In the console screen, we can see that the script is running through the dataset, outputting information to assist with any possible debugging. At the end of the analysis, there's one more step required to generate the report. Because this is a Markdown file, we will need to execute the knit command to convert the intermediate output into a PDF. The render window will display a few processing steps as the document is converted into a PDF entitled Florida Crop BMP QA/QC Single Dataset. When completed, the report will appear in the working folder for review. Generating a QA/QC report for a BMP dataset is a straightforward process. However, the QA/QC tool is designed to verify dataset completeness, not to detect data errors. Researchers should conduct their primary data checks prior to uploading. Continuously updating the BMP workbook and rerunning the QA/QC script throughout the research process helps ensure the accuracy of the records and minimizes the risk of errors spreading across worksheets. Florida's agriculture industry depends on up-to-date best management practices reinforced by reliable research. By following the FAIR principles and building on the ICASA data standards, the Florida Crops BMP data template will generate valuable and accessible datasets for ongoing BMP research, ultimately giving farmers the guidance they need to remain competitive while protecting Florida's natural resources. This 4-part training module provides an overview of the structure and use of the Florida Crop BMP data template. For questions and comments, users can consult the other instructional videos in this series or follow the links for additional information on the FDAX website.